light reaction is having 3 steps. Concerned to 3 steps, definitely there are many NEET questions. The light reaction is having the first step, photo-oxidation of chlorophyll molecule. First one. Already we have studied about LHC, reaction light harvesting complex, which is having a reaction center having chlorophyll A and is surrounded by chlorophyll B, xanthophyll and carotenoids. Those 3 together named as antennae. Those are incapable to, they are capable to trap radiant energy of sun and supply to the reaction center, chlorophyll A. Chlorophyll A, when gets that energy, solar energy from antennae, it goes to higher level. It goes to higher level. And that chlorophyll molecule loses electron and becomes excited. And that ejected electron is received by many electron acceptors. It happens in each and every LHC. Whenever the light falls on light harvesting complex, antennae trap that light and supply to the reaction center. And now the reaction center, by getting solar energy, it goes to higher level and ejected out the electron. Once the electron is ejected out, it becomes excited. In chemistry, we used to mention, instead of excited, oxidized. And that ejected electron is received by many electron acceptors. This is photo-oxidation of chlorophyll molecule or photo-excitation of chlorophyll molecule. And you know about 2 LHC, photosystem first and photosystem second. If it is photosystem first, where chlorophyll A700 is there, and photosystem second, where chlorophyll A680. This is about photo-excitation of or photo-oxidation of chlorophyll molecule. Whenever the light falls from antennae, that radiant energy comes to chlorophyll A and it goes to higher level, loses electron, becomes excited, and ejected electron passes through many electron acceptors. And now second one is photolysis of water. Photolysis of water, that is light governed splitting of water, or photoionization of water is otherwise said to be photoionization of water. See here, water in presence of light and it splits and giving electron, oxygen and protons. To balance the equation, let me take 2 molecules of water so that 2O2 means O2 and there are 2 H2 means totally we do have 4 electrons and 4 protons as well. 4 electrons and 4 protons together, 4H. This is balanced. It is the equation for photolysis of water. Equation for photolysis of water. Which of the following is an equation for photolysis of water? Even they can give like this also. And H2O in presence of light or photons, 2 electrons, O and 2 protons. If they give this also is right, If they give this also is right. To make O2, I have taken 2H2. Otherwise, whenever the water splits, it gives 2 electrons to protons and oxygen. It is photolysis of water. Remember, this photolysis of water is in association with photosystem second. Photolysis of water is in association with, that question they can ask, is associated with photosystem second. This is second step as well. And third is photophosphorylation. Photophosphorylation. What is phosphorylation? That is nothing but formation of ATP from ADP and inorganic phosphate. It is phosphorylation. But I have written photophosphorylation. Photophosphorylation means light governed formation of ATP from ADP and inorganic phosphate. That is said to be photophosphorylation. And we do have two types of photophosphorylation. First one, cyclic photophosphorylation. And second one is non-cyclic photophosphorylation. Remember, phosphorylation means formation of ATP from ADP and inorganic phosphate. Photophosphorylation means light governed formation of ATP from ADP and inorganic phosphate. It is having two types. One is cyclic photophosphorylation. What is cyclic photophosphorylation? It is one where the ejected electron from excited chlorophyll molecule passes through many electron acceptors, but it comes back to its own chlorophyll molecule. It comes back to its own chlorophyll molecule. That is cyclic photophosphorylation. For the cyclic photophosphorylation, only photosystem first is required. It means the reaction center is P700. And now non-cyclic photophosphorylation. It is one where the ejected electron from excited chlorophyll molecule passes through many electron acceptors, but it never comes back to its own chlorophyll molecule. It never comes back to its own chlorophyll molecule. In first one, cyclic, the ejected electron from chlorophyll molecule passes through many electron acceptors, but it comes back to its own chlorophyll molecule. Second one, non-cyclic photophosphorylation. Here the ejected electron from excited chlorophyll molecule passes through many electron acceptors, but it never comes back to its own chlorophyll molecule. If I draw the diagram, we can easily understand these two. Even we can discuss the questions concerned, the NEET questions concerned to cyclic and non-cyclic photophosphorylation. And for non-cyclic photophosphorylation, both photosystem first and photosystem second are required. Photosystem first means reaction center is P700. Photosystem second means P680. These also the questions, NEET questions. In graph only, in diagram only, I will explain about that. Let us see the diagram. Yes, I have just mentioned what is cyclic photophosphorylation. Cyclic photophosphorylation is one where when photons fall on the chlorophyll molecule, it goes to higher level loses electron, becomes excited, and ejected electron from excited chlorophyll molecule passes through many electron acceptors, but it comes back to its own chlorophyll molecule. It comes back to its own chlorophyll molecule. On the way, there is a formation of ATP from ADP and inorganic phosphate. Here, there is a cyclic flow of electron. The electron which is ejected from the chlorophyll molecule comes back to its own chlorophyll molecule as well, and is cyclic photophosphorylation. And what are the NEET questions they can ask concerned to this? In cyclic photophosphorylation, which of the following are formed? Our answer is 1-ATP. Remember, in cyclic photophosphorylation, there is a formation of 1-ATP. This is one NEET question. And second is, which of the following LHC is involved in cyclic photophosphorylation? That is photocystin first. In cyclic photophosphorylation, which chlorophyll molecule is a reaction center? Chlorophyll A700. It is said to be P700. P700 means a reaction center having a chlorophyll A700. Then, this is taking place on stroma lamellae. Here, three things you should remember with respect to this. In cyclic photophosphorylation, just 1-ATP is formed. Cyclic photophosphorylation involves photocystin first. As photocystin first is involved, P700 is there, and this cyclic photophosphorylation takes place on stroma lamellae or intergranule lamellae. These three things you must remember. That's all. This is a schematic representation of non-cyclic photophosphorylation. Just now I mentioned the definition for non-cyclic photophosphorylation. The electron from ejected electron from excited chlorophyll molecule passes through many electron acceptors, but it never comes back to its own chlorophyll molecule. Here, in a non-cyclic photophosphorylation, you should remember the questions which are raised in NEET. For non-cyclic photophosphorylation, both photocystin second and photocystin first are involved. This is photocystin second and photocystin first. When photons fall on photocystin second and photocystin first, simultaneously the chlorophyll molecule 680 and 700, they go to higher level. They go to higher level. Once they go to higher level, they ejected out the electron. Once electron is ejected, they will be excited. They will be excited. Just mentioned in biology, we have used the term excited, but in chemistry is oxidized. When electron is ejected, these chlorophyll molecules are excited or oxidized, and they are received by electron acceptors. Received by electron acceptors. And the electron ejected from photocystin second passes through electron transport system, and the electron from this photocystin second, it will come to photocystin first. It comes to photocystin first. It never comes back to its own chlorophyll molecule. Electron from photocystin second, it comes to photocystin first. It never comes back to its own chlorophyll molecule. And while electron, which is moving from electron transport system, 1 ATP of energy is formed. 1 ATP of energy is formed. And now, the electron ejected from photosystem first is received by electron acceptor, and the same electron is used to reduce NADP into NADPH2. That electron is used to reduce NADP into NADPH2. Another assimilating power is formed. But this electron acceptor can provide electron, but protons, those protons are provided by a photolysis of water. Photolysis of water, where protons are generated, they are used by NADP. Those protons and electrons are used. Now NADP becomes NADPH2. And then the electron from photosystem second, it has come to photosystem first. But it has to get the electron, It gets electron from the electrons which are formed during photolysis of water. The electron formed during photolysis of water. And if you remember this schematic representation, you can answer for many NEET questions. Already I have mentioned non-cyclic photophosphorylation requires photosystem second and photosystem first. And it is taking place on grana. It takes place on grana. And then they may raise many questions. What is the outcome of this non-cyclic photophosphorylation? 1 ATP and 1 NADPH2. But in cyclic photophosphorylation, just 1 ATP. And in non-cyclic photophosphorylation, 1 ATP and 1 NADPH2 are formed. Then, they can make many NEET questions. In non-cyclic photophosphorylation, the excited chlorophyll 680 comes to ground level by getting the electron from the electron formed during photolysis of water. And the next question, the excited chlorophyll A700 of photosystem first, it comes to ground level by getting the electron, that is from photosystem second. It gets electron from photosystem second. And the electron of photosystem first is used, next question, for the formation of NADPH2. The electron from photosystem first is used to reduce NADP into NADPH2. Remember these 3 points where they can make the NEET question. The excited P680 comes to ground level by getting the electron from photolysis of water. And the excited chlorophyll 700 of photosystem first comes to ground level by getting the electron from photosystem second. Photosystem second. And the electron of photosystem first, it is used for to reduce NADP into NADPH2. And remember the very important role of photolysis of water in a non-cyclic photophosphorylation. How? It provides protons to reduce NADP into NADPH2, and also it provides electron to the photosystem second to come to the ground level. This is a non-cyclic photophosphorylation. And here I have pinpointed the NEET questions wherever they ask as well. And even here one more question they can ask. Photolysis of water is in association with 1. Non-cyclic photophosphorylation, 2. Cyclic photophosphorylation. The answer is associated with non-cyclic photophosphorylation. Yes, these are the cyclic and non-cyclic photophosphorylation graph, and this non-cyclic photophosphorylation schematic representation, it is said to be Z scheme. Z scheme. And even in your NEET, they can give the graph where instead of mentioning, they don't write this. This is, they mention A. Here also they don't write anything. This is B. And then C, D. By giving this, in the answer they will give decoding. A, what is that? A is equal to P680, B is equal to P700, C is equal to NADPH2, and D is equal to ATP. And both the assimilating powers which are formed during non-cyclic photophosphorylation, both of them are not formed during cyclic photophosphorylation. It's about the light reaction spell. While discussing about photophosphorylation, whether it is cyclic photophosphorylation or non-cyclic photophosphorylation, where there is a formation of ATP. How this ATP is formed? It was explained by the hypothesis, a theory named Chemiosmotic hypothesis. They have explained the formation of ATP. Here it takes place in thylakoid membrane. This circular is thylakoid membrane, and it is inner membrane and is outer membrane and is inner membrane. And inner membrane encloses the cavity inside named lumen. It is lumen. And outside, totally, this is stroma. Stroma of chloroplast, and is a thylakoid membrane that is there in grana. In thylakoid membrane, photocystin second and photocystin first are connected by electron transport system. In non-cyclic photophosphorylation, I have mentioned electron transport system, that is having cytochromes. What are cytochromes? Cytochromes are proteins. They are electron acceptors. They receive energy-rich electrons and transfer to the next cytochromes. They are designated as cytochrome A, A1, A2, A3, B1, B2, B6, like this. There are many cytochromes are there. They are nothing but protons, and they are electron acceptors. Cytochromes belonging to which group of organic compounds is a NEET question? Cytochromes belonging to proteins, and the cytochromes are electron acceptors. In the stroma, there are protons. And in the non-cyclic photophosphorylation, photosystem second, from photosystem second, electron comes to photosystem first. Through the electron transport system, that electron transport system is having cytochrome B and F complex. B and F complex. whenever the electron from photosystem second comes to cytochrome B and F complex, the cytochrome B and F complex, I consider it as A. When this electron comes to cytochrome B and F complex, that is considered as A. Whenever the electron comes, proton from stroma are also coming. This electron comes from photosystem second and a proton comes from stroma, leading to the formation of AH2, reduced cytochrome B and F. To balance this, 2 electrons, 2 protons. It gets reduced. Whenever, remember this point, whenever electrons come to cytochrome B and F, I consider B and F as A. Whenever electron comes, proton from stroma will come to that complex, so that A gets reduced into AH2. And now this AH2 splits into A back, 2 electrons, 2 protons and 2 electrons. Then first AH2 is formed, once again is splitted into A, that B and F complex, protons and electrons are formed. The electrons which are generated, they will go to photosystem second, and the protons, they are pumped into lumen. Those protons are pumped into lumen. Whenever electron is coming from photosystem second to that cytochrome B and F complex, proton from stroma will also come and A gets reduced to AH2. A is nothing but cytochrome B and F complex. And now this splits. A is regenerated, means B and F are regenerated, and electrons which are formed go to photosystem first, and protons are pumped into lumen as well. Protons will come to lumen, and electrons which are received by photosystem first are used to reduce NADP into NADPH2. This you could see in that non-cyclic photophosphorylation, and where NADPH2 are formed continuous, while electrons moving from photosystem second to photosystem first through cytochrome B and F, protons are pumped into lumen, so that the concentration of protons becomes more in the lumen than stroma, so that proton gradient is created. Gradient in the sense, protons which are more accumulated in the lumen, less in stroma. As the proton gradient is created, that makes proton motive force. Due to that proton motive force, the more protons which are there in the lumen, they are pumped out into the stroma through ATP synthase. This ATP synthase is having 2 parts. One is F0, another one is F1. And this ATP synthase is considered as F0 F1 complex. That F0 is just like stalk and F1 is an outgrowth, so that protons which are there in the lumen are pumped out through the F0 F1 complex. Once those protons are pumped into the stroma, the maximum energy is released. That energy is used by ADP, so that ADP can react with inorganic phosphate, hence the formation of ATP. In this way, synthesis of ATP takes place. Remember, this here also they can give A, B and C. They don't give anything. Decoding you have got to do. This is photosystem second, cytochrome B and F, and is photosystem first. And decoding can be done to ask any question concerned to this. And remember, two parts of ATP synthase are F0 and F1. And see here, proton concentration increases in the lumen. That is due to the flow of electron from photosystem second to photosystem first through cytochrome B and F. To reduce that cytochrome B and F into reduced cytochrome B and F, and once again it splits, electrons are pushed to the photosystem first, protons are pumped into lumen. Due to that, proton concentration in lumen increases as compared to stroma, so that proton gradient or gradation is created. It means more protons in the lumen and less in stroma. Due to proton gradient, a proton motive force is created, so that protons from the lumen are pumped out into the stroma through F0 F1 complex. While moving through, maximum energy is released, that is used by ADP, then it reacts with inorganic phosphate in presence of ATP synthase enzyme, leading to the formation of ATP. And the electrons which have come to photosystem first, that you have seen in non-cyclic photophosphorylation. When the electrons which have come here, they are used to reduce NADP into NADPH2. It's about the formation of ATP, which is explained by Chemiosmotic hypothesis. And our NEET questions, they can give this structure in which everything will be coded, A, B, C, and then here D, E, everything. Your work and right answer where they can give, A is photosystem second, B is cytochrome B and F, and C is photosystem first, and D F0, E F1, like this. They can make the question. Even concerned to this, proton motive force is created by proton gradient between lumen and stroma. And proton gradation motive force is created when more protons in the lumen than stroma. Then only protons are pumped out. Protons come in and they are pumped out. That's all. Already we have discussed about light reaction, which is taking place in grana. The net products are formation of ATP and NADPH2. These are assimilating powers. Only one of the substrate, water, is used and is oxidized to give O2. Now it's time to discuss about dark reaction, and the dark reaction is taking place in stroma. The second step is said to be dark reaction because it is independent of light. Even is also named as biosynthetic phase, because there is a synthesis of glucose during dark reaction. As it is independent of light, it doesn't mean it is taking place only during night. No. Dark reaction, it takes place during day and night and is taking place in stroma. Even it is also named as Kelvin cycle. As Mr. Kelvin found out the cyclic pathway and also named as Blackman's reaction. Here in a stroma, CO2 is there and the ATP and NADP H2, the assimilating powers which are formed during light reaction are also have come to stroma. Here that CO2 has to get reduced into carbohydrate. Kelvin explained the cyclic pathway in which that CO2 is fixed in the reaction and there would be a formation of glucose and that takes place. That Kelvin cycle means dark reaction takes place in 3 steps. Those 3 steps are, first one, CO2 fixation, otherwise said to be carboxylation. Second one is reduction. And third is regeneration of RUBP. That RUBP is nothing but ribulose biphosphate. Ribulose biphosphate. A 5-carbon compound which is there in the stroma that fixes CO2. The Kelvin cycle takes place in 3 steps. In first step, CO2 fixation or carboxylation takes place. It means CO2 is fixed by this RUBP, and in second step, reduction, and third step, how much RUBP which are used to fix CO2 are regenerated. If I draw that cyclic pathway, we can understand, even we can get as many as NEET questions concerned to this Kelvin cycle. This is a Kelvin cycle. Here you can see the cyclic pathway as well. The dark reaction takes place in 3 steps: carboxylation, reduction, and regeneration of RUBP, and is taking place in stroma. In a stroma, there are many RULP. They are nothing but ribulose monophosphates. They all become RUBP. And this RUBP, it is a 5-carbon compound. It is a 5-carbon compound having 2 phosphate groups. This is capable to fix carbon dioxide in dark reaction. There would be a NEET question. In dark reaction, carbon dioxide is fixed by RUBP, ribulose biphosphate. The fixing of that CO2 by RUBP is named as CO2 fixation or carboxylation. Once one molecule of RUBP fixes one CO2, there will be a formation of 2 molecules of 3-phosphoglyceric acid. What this 3 indicates? The phosphate group is attached to 3rd carbon of glyceric acid. See here, in RUBP 5 carbons, CO2 having 1 carbon total number of carbons are 6. When that RUBP fixes CO2, there will be 2 molecules of 3-phosphoglyceric acid are formed. Total carbon here 5 plus 1. 2 molecules of these are formed means this 3-phosphoglyceric acid is having 3 carbons. And these are 2 molecules means 2 into 3, 6 carbons, where 5 plus 1, 6 carbons. 2 molecules of 3-phosphoglyceric acid is formed. And it is the first formed stable compound in dark reaction. Hence, this Kelvin cycle, it is said to be C3 pathway. This Kelvin cycle is said to be C3 pathway because the first formed stable compound in the Kelvin cycle is 3-phosphoglyceric acid, which is having 3 carbons. Hence the name C3 pathway. And for this Kelvin cycle, it is another name, C3 pathway. Why this is said to be C3 pathway? The first formed stable compound in Kelvin cycle or dark reaction is having 3 carbons, which is none other than 3-phosphoglyceric acid. Then in C3 pathway, which compound fixes CO2? RUBP. And the first stable compound formed is 3-phosphoglyceric acid. 3-phosphoglyceric acid. Sometimes 3-phosphoglycerate. It can be said 3-phosphoglycerate. When glycerate comes, glyceric acids. And now, but in dark reaction, how many CO2 are used? 6 CO2 are used. Here 1 RUBP fixed 1 CO2 forming 2 molecules of 3-phosphoglyceric acid. But in our reaction 6 CO2 are required. Then to fix 6 molecules of CO2, we need 6 molecules of RUBP. To fix 6 CO2, we need 6 molecules of RUBP. When 1 RUBP fixes 1 CO2, 2 molecules of 3-phosphoglyceric acid are formed. Now, 6 molecules of RUBP fix 6 CO2. How many 3-phosphoglyceric acid will form? 12. 12 molecules are formed. Now 6 molecules of RUBP fixes 6 CO2. There will be the formation of 12 molecules of 3-phosphoglyceric acid. Okay? Then, and these 3-phosphoglyceric acid, they are totally 12 molecules. They get reduced in presence of ATP and NADH2. To reduce 1 3-phosphoglyceric acid, we need 1 ATP and 1 NADPH2. But how many of them are there? 12 molecules. So that we need 12 molecules of ATP and 12 molecules of NADPH2 are required. Hence, these 3-phosphoglyceric acid are reduced, leading to the formation of 12 molecules of triose phosphates. 12 molecules of triose phosphates. And these are also 3 carbon compounds. 3 carbon compounds. 12 molecules. In which just 2 molecules are used to generate 1 molecule of glucose. It is having 6 carbon. In 1 triose phosphate, 3 carbons are there. We need 2 triose phosphates to form a 6 carbon glucose. and then gets polymerized and forming a polysaccharide starch. But how many of them are there? Totally 12. But 2 are used to generate glucose in the reaction. But 10 are remained. 10 triose phosphates are remained. These 10 triose phosphates, they are used to regenerate 6 molecules of RUBP. How many of them are? 12 totally, in which 2 are used, and 10 molecules are used to generate that. See here, to generate one RUBP, one ATP has to used. But we need 6 molecules, means 6 ATP must take part, leading to the formation of 6 ADP. And 6 molecules of RUBP are formed. You just balance the carbon. In a total 12, 2 are used to generate glucose. 2 3 carbon compounds are used, means 6 carbon glucose is formed. And 10 molecules are remained, 3 carbon. 10 into 3, 30 carbons. And see here, there are 6 molecules of RUBP. They are 5 carbon compounds. 5 into 6, 30 carbons. 10 molecules with 3 carbon compounds, they are 30, and 6 molecules with 5 carbon, 30 carbon atoms as well. So that from 10 triose phosphate, there is a regeneration of 6 RUBP. It is taking place continuously. CO2 is fixed by RUBP, 3 phosphoglyceric acid are used, ATPs and NADPH2, which are formed in light reaction, have come to the stroma, and they are used to reduce 3 phosphoglyceric acid, forming triose phosphates. From triose phosphates, glucose and starch, our required product is formed, and remaining triose phosphates are used to generate RUBP. And cyclically, this process is taking place. This is C3 pathway. We can raise now NEET questions. It is said to be C3 pathway because the first formed stable compound in Kelvin cycle is having 3 carbons, which is none other than 3 phosphoglyceric acid. There are 4 questions, multiple choice answers are given. One among them will be this. And in the C3 pathway, the first formed stable compound is 3-PZA. They may give 3-PZA. I have given an elaborated form. 3-phosphoglyceric acid, that is 3-PZA. And in the C3 pathway, which of the following compound fixes the CO2? RUBP, which is a 5 carbon compound. That also they can ask. And as usual, whenever the scheme or cycle is there, coding can be done. In RUBP, where they can give A, here B, and C. And as per as steps, first, second, and third, here they can give X, Y, and Z. If they wish to ask the steps, here they can give the compounds names will be given, but here they can give X, Y, and Z. Sometimes, if they wish to ask the compounds, they will write carboxylation, reduction, and regeneration of RUBP, but here they can give code X, Y, Z, like this, so that you should remember those compounds. All these schemes and cycles you should remember, because decoding they will give A is equal to RUBP, B is equal to 3-phosphoglyceric acid, C is equal to triose phosphate. Like this, they can ask the question as far as NEET is concerned. And for this, we can make the complete reaction. How many RUBP are used? 6 RUBP. They fix 6 CO2. How many 3 phospho? These are intermediate products. The product formed is C6H12O6 and 6 RUBP are used. And one more thing I did, 6 RUBP fixes 6 CO2. Total number of ATP used in this, here 12, here 6, 18 ATP. How many NADPH2 are used? 12 NADPH2. And leading to the formation of one molecule of glucose. And then those 6 RUBP are regenerated. Then in 18 ATP, 18 ADP are formed. 12 NADPH2. As they give hydrogen, 12 NADP plus. This is the equation for dark reaction. This is the equation for dark reaction. They will give many equations. In which of the following is the equation for dark reaction? 6 RUBP fixes 6 CO2, 18 ATPs and 12 NADPH2 are used to form one molecule of glucose, and the 6 RUBP are regenerated. From 18 ATP, 18 ADP are remained. From 12 NADPH2, 12 NADP plus are remained. That also they can ask. Even in a simple manner, in a C3 pathway, number of ATPs used, 18 ATPs. In C3 pathway, number of NADPH2 used, 12 NADPH2. And number of RUBP used and regenerated, 6 are used, 6 are regenerated. That they can make multiple choice. 6 are used, 9 are regenerated. Like this they can give. 6 RUBP are used and 6 RUBP are regenerated. And even this is said to be C3 pathway because the first formed stable compound is 3-phosphobasic acid. But in some plants, mainly graminate members like cereals, sugarcane, there is a C4 pathway. As the name indicates C4 pathway, as usually we can say in C4 pathway, the first formed stable compound is having 4 carbons.